Next experiment, identification of marine planktons. Materials required: plankton net, dissection microscope, petri dish, glass slides, etc. With the help of fishing boat, we collected the marine plankton using an ordinary plankton net. The collected sample was transferred to a glass bottle. The organisms were narcotized and killed by 5% formalin and diluted. Then the sample was taken into centrifuge and studied. Each type of animal is transferred into different container. The plankton were stored in various taxonomic groups. It was done with the help of zero water, brush, handlers, and dissection microscope. They contain some microscopic forms, large organisms, and larger larval stages. The organisms Zoea, Mysis, adult, and larvae, Lucifer, copepod, etc. were analyzed in collection. Conclusion: the majority of plankton animals belonging to the class crustaceans, copepod, are most important and serve as the chief food for commercially important fishes. Special adaptations are found among planktons organisms to tide over unfavorable physical conditions. Based on the salinity and temperature of water, different community of plankton animals that are analyzed found that different levels of water, vertical migration is seen around certain groups of plankton organisms. Then result: the following are important planktonic animals that are analyzed and studied in marine conditions. They are crab, zoea, copepod, lucifer, mysis, and sagitta. Crab zoea. A zoea is a larval stage. Zoea is a larval stage in the development of crab and other decapod crustaceans. Crab zoea larvae has a strong and almost spherical carapace to which the legs it was attached for swimming. Legs are absent for the long and extended back. Then they are often long and prominent rostrum always pointed downward, but can occasionally be absent. Crab zoea larvae legs are absent and the long and extended back, the often long and prominent rostrum always pointed downward, but can occasionally be absent. Crab zoea larvae usually have several other outgrowths on their body. Among them, a very prominent vertical outgrowth is back. Phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda. Phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda. Once again, phylum Arthropoda, phylum Arthropoda, subphylum Crustacea, class Malacostraca, order Decapoda. Copepods are free-swimming commensals and parasites. The body is small, elongated, and composed of head, thorax, and abdomen. Carapace is reduced or absent. Lateral compound eyes are absent. A single median eye present. Copepods are free-swimming commensals and parasites. The body is small, elongated, and composed of head, thorax, and abdomen. Carapace is reduced or absent. Lateral compound eye absent. A single median eye present. Antennule and antennae will be developed and used for swimming. Mandible may bear palps. The typical 5 pairs of thoracic appendages, of which first uniramus and last biramus. Sometimes 6th pair of vestigial appendages and is a pair of caudal style. The first thoracic or the second also in some cases segment is fused with head to form cephalothorax. The genital aperture on the last thoracic segment are here between and the genital segment. Once again, I am going to read the copepod. Copepods are free-swimming commensals and parasites. The body is small, elongated, and composed of head, thorax, and abdomen. Copepods are free-swimming commensals and parasites. The body is small, elongated, and composed of head, thorax, and abdomen. Carapace is reduced or absent. Lateral compounds are absent. A single median eye present. Antennule and antennae will be developed and used for swimming. Mandible may bear palps. The typical 5 parts of thorax appendages of first uniramus and last biramus. Sometimes 6th pair of vestigial appendages and is a pair of caudal style. The first thorax or the second also in some cases segment is fused with head to form cephalothorax. The genital aperture on the last thoracic segment aligns between as the genital segment. Final Arthropoda, Gastropoda, Maxillopoda. Class Arthropoda, Subphylum Crustacea, class Maxillopoda. Arthropoda, Crustacea, Maxillopoda. Crabzoia, dorsal spine. Carapace, compound eye. Dorsal spine. In case of Crabzoia, dorsal spine, carapace, compound eye and abdomen can be seen. Some rostral spine is also there. Copepod, antennae, setae, eye. Antennae, setae, eye, thorax, abdomen, egg sac and caudal. Ramus, copepod. Antennae, setae, eye, thorax, abdomen, egg sac, caudal, ramus. Then Lucifer. Lucifer is a little known and degenerate genus of prawns. It has a long body but many fewer appendages than other prawns, with only 3 pairs of periopods remaining, all without claws. It also bears no gills. The female, uniquely among prawns, carry the fertilized egg in her periopods until they are ready to hatch. The head is extremely elongated and bears long antennae and eye with a stalk. The 5th periopod is absent on vestigial. Luminescent organs are absent, but luminescent cells are present in telson. Lucifer is a little known and degenerate genus of prawns. It has a long body but many fewer appendages than other prawns, with only 3 pairs of periopods remaining, all without claws. It also bears no gills. The female, uniquely among prawns, carry the fertilized egg on her periopods until they are ready to hatch. The head is extremely elongated and bears long antennae and eye with a stalk. The 5th periopod is absent or vestigial. Luminescent organs are absent. Luminescent cells are present in telson.